Welcome back to Crosstalks. For those of you joining us for the first time, Crosstalks is a collaboration between Stockholm University and KTH, the Royal Institute of Technology. A quick reminder, you do have the unique opportunity to ask questions directly of our studio guests towards the end of this show. Simply call us on Skype, where our name is Crosstalks TV. And do discuss today's topics on Twitter, where we call ourselves Crosstalks TV. Our hashtag is Crosstalks. The Ebola outbreak keeps spreading despite efforts to contain the virus with curfews and closed borders. How should we handle the threat of contagion in our interconnected world? What technologies are available? How can we predict and monitor similar outbreaks? Which political and social methods are most effective for minimizing the spread of disease? Joining us today in the studio are Rob Daniels, Assistant Professor at the Department of Biochemistry and Biophysics, Stockholm University, Lars Skog, Ph.D. in Geoinformatics at KTH Royal Institute of Technology, and Pieter Trapman, Researcher at the Department of Mathematics, Stockholm University. Please give our guests a warm round of applause. Rob, you study the influenza virus on a molecular level. What are the aspects that you focus on in your research?
So like you said, we study influenza, and like all viruses, influenza wants to enter cells, replicate, and then spread. And influenza has an envelope or a membrane that surrounds it, and it contains two proteins. One is hemagglutinin, which facilitates entry, and the other is neuraminidase, which facilitates release and spreading. And what we've been focusing on is neuraminidase and how it's changed or evolved since 1918 to 2009 due to the fact that it was one of the two changes in the virus that led to the 2009 pandemic.
Wow, very good. Can you also help us understand a little bit what we would use this understanding for?
Basically in terms of how viruses are evolving to transmit, and how they're responding to vaccines, and basically what's happening in terms of the human virus. Is it becoming less lethal? Is it becoming more lethal? And why are we getting the particular changes in the virus that we're seeing on a sequence level in terms of mechanisms and how and what the end function is? I'm
immediately a little bit worried. I will just ask one more follow-up before I let the others in. Is the human influenza virus getting worse or less dangerous?
I mean, just from a sheer number standpoint, it's getting less. If you think of the 1918, we killed, I think, 50 to 100 million people. And then if you come back to the H2N2 virus back in 1957, somewhere between 5 and 10. And then the H3N2 virus that entered in 1967 would be around 1 million. And we're in the 100,000s these days. So just by numbers, you would say, this is per year, you would say that we're less.
Yeah, that's comforting. Lars, you have developed a method that medical professionals can use to visualize the geographical distribution of diseases over time. Could you talk us through what it is and how it works?
Yeah, I'll try. At our division at Geoinformatics, we deal with geographic information science, or GIS, and that is about combining geographic information, storing it and analyzing it and visualizing it. The typical sources for that are satellite imagery or aerial photography, or could also be ordinary maps in digital format. The nice thing about GIS is that say position is a common denominator for almost any kind of information. And that means that you can combine data from different sources with the geographic information that you have stored in advance. I have looked at the spread of pandemics, and doing so I have used different methods or a methodology that starting with data preparation and geocoding, data spatial analysis, spatial modeling, and or network analysis and visualization of the data gathered and analyzed. And then with the case or case data from infections, then the tricky thing is data preparation, because the quality of the case data varies a lot.
What does that mean? It's databases and I guess, hospital records, these kinds of things?
Yes, could be hospital record from influenza-like symptoms, not necessarily the influenza in itself, and it could also be laboratory verified cases, and that was the case for the 2009 influenza. But the other ones I've studied, then it's more like symptoms.
And what comes out the other end? It's a map, essentially.
It's a map, but this, say, as you have time as a parameter, you can animate sequences over a period. Say, you can visualize what it looks like week by week, and this way, epidemiologists get a clear view of what has been happening, and that also give a basis for discussion on how future pandemics will behave.
So we've talked about molecular understanding, and this is a sort of geographical understanding. Now, Pieter, you work with mathematical modeling of infectious diseases. What can we learn by looking at outbreaks through statistics that we wouldn't learn otherwise?
That's a good question. So mathematical modeling can teach us quite a lot. It already started in the 18th century when Bernoulli evaluated the effect of smallpox vaccines, which were not without danger back then to use the vaccine. But he showed that if people use the vaccine, there will be on the whole less victims of smallpox than if nobody uses the vaccine. So the computations are pretty clear. Well, there is quite some maths behind the work the tools that Lars is using as well.
Yeah.
But in general, like in mathematical modeling, the holy grail for epidemiologists is kind of R0, which is the average number a typical infected person will infect if he gets infected himself. So if that number is below 1, then probably the disease won't get big, or the epidemic won't get big. If it's above 1, it might get big. And this R0 is pretty robust. You can use it for all kinds of models, including which include social behavior as well. So if you want to analyze the control strategies, it's very important that you know how to use mathematics to compute this R0, even after control strategies have taken effect.
So then you would retroactively do the calculation to evaluate whether it's worked or not?
Yeah, and because of the mathematics, we know that it's pretty robust to do those calculations. So we have a pretty good idea what we need to do. Of course, people sometimes behave strange, but sometimes we can also identify adverse effects. There is the case of rubella vaccination, where, well, if you get enough people vaccinated against rubella, then the effective R0, so the average number of people which will get infected by a typical infected individual, will be below 1. But if you vaccinate not quite enough, then the main effect is that the epidemic will, or the disease will spread slower, which actually cause that people contract the disease later in their life. And rubella is pretty harmless if it comes to children, which usually get the infection. But if you vaccinate not quite enough people, then many or the number of pregnant women who will contract the disease and will get sick of it will actually increase.
This is an aside, but I think that this is a pretty solid argumentation against these people who are stopping vaccines now based on, I don't know what, but on unscientific principles, we can say. Yes.
Yeah, or in cases where you cannot reach enough of the population,
well, yes,
then you might consider not starting vaccination against rubella at all.
Oh, that's fascinating. You were both humming and nodding at this holy grail of the average number of infected patients. Would you like to add to that? For instance, why Holy Grail sounds like something that's very difficult to achieve.
I mean, basically, when Peter's talking about the R0 number, he's referring to transmission. And essentially, that's one of the things when you look at any infectious agent, you want to identify it, and then you want to determine what mode of transmission is actually occurring. People always talk about airborne transmission, and with Ebola, people talk about direct contact transmission. And these types of things are what change these values. Right, so for sure to be able to predict them, is completely advantageous to understand what's going on and potential danger.
Lars?
I think I pass here.
Okay, but I thought it was interesting to see also because you display an intellectual curiosity about epidemics as well. That I mean, of course, they're terrifying and awful, but they do seem to be a little bit exciting as well, which is fun to see. Rob, how do you define a potential pandemic pathogen?
I think if we just start from a basic analogy, everyone in the audience here today is exposed to millions of billions of potential pathogens just by breathing. Yet we all rarely get sick, and that's because we as humans have a wonderful immune system that over time gets exposed to different infectious agents, and then it gets trained so that when we see it a second time, we're less likely to develop symptoms. Pandemic pathogens, which could be bacteria or viruses, are lethal, which by definition, or highly lethal in humans, which by definition means the human population as a whole has basically not been exposed to this infectious agent before. So that means that the bacteria or the virus has to have come from another source that's not of human origin, and usually that's animals. And that's for the case of influenza, that would be, it's at least thought to be birds, for instance. And so if that virus crosses into humans, it can be lethal. It becomes a potential pandemic threat. But once it gains the ability to transmit from human to human, then you have a pandemic agent. Yeah.
Could you just give us some historic examples of what we're talking about here?
Sure. In terms of one of the most famous ones would be the Black Plague back in the late 1300s, which was a bacteria housed in the fleas on rats that spread very slowly through Europe and the rest of the world. And then in 1918, we had the influenza pandemic, or Spanish flu. And then as we've come more recent, we've had more what we'd probably classify as potential pandemic threats, and that would be Ebola, which was first discovered in 1976, and more recently resurfaced along with MERS-CoV, or Middle Eastern Respiratory Syndrome coronavirus, in 2012.
So, I'm sorry if this is such a layman's question, but you said, of course, that the danger is something that the population hasn't been exposed to before. Is this why influenza is less dangerous? Do we as a population get better at handling it, or is it just that our medical sciences have also advanced so much in the last century that we can keep it at bay?
I mean, there's two things. One is vaccination, for sure. First pandemic, 1918, no vaccines. I think vaccines were introduced in the late 1950s. So hence that would also lead to the decreased numbers I was talking about earlier. But in terms of flu, there's always the reservoir of the different influenza types is quite substantial. Like recently, they've even found strains that we haven't identified before in bats. So if these strains that we haven't seen develop the capacity to jump the species barrier into humans, then they can very well cause a new pandemic, because we don't have a vaccine for those.
Oh yes, of course, yeah. So we do get new influenzas as well. If it was only the old one and it didn't mutate, then we could solve it. But because they do, there it is. So Lars, you have used your model to explore the relationship between geography and disease distribution in some of these past epidemics. What does one learn?
Well, I have studied 3 pandemics. You could say it was the Russian flu in 1889 to 90, the Asiatic flu in 1957 to 58, and the swine flu now in 2009. And And also I've been looking at the Black Death. I have been fortunate enough to work with Swedish data, as there is a lot of data available really in Sweden and on population, and there's a lot of statistics that you can analyze, and except for the Black Death, of course, because then there were no data at all. So I had to model what it could have looked like, knowing something about the disease etiology, which I think is an extremely important factor when you're thinking, is a pandemic dangerous or not? You have to know what causes it. And the people in the Middle Ages, they had no idea about the bacterium or the rat fleas or anything. But it had a very dramatic development. And in Sweden, for instance, 30% of the population was killed in 1350 at the first outbreak. And then there were a few more outbreaks, but it took 300 years until the Swedish population reached the level it had in 1349, 1 million people. And if you compare that to Ebola today, there are, I learned yesterday, about 15 per mil of the population in Liberia have been affected, but the Black Death was 2,000 times as severe.
And not so much because of the mortality that comes with the disease, but because we know so much more now.
No, I think that has to do with, as I say, the mortality of Ebola. I'm not an epidemiologist, so I should not speak out too loud. But
you can hear.
But still, you know it's transferred via body fluids, and in West Africa, people seem to have the tradition to be very close to the deceased corpse. They wash them and so, and it's easy to that the contagion is transferred to new humans, and also for the healthcare workers that is the case. I would also say that the situation is there. We will hear more about that later, so I'll stop there.
Well, let's do that right now. I think we should bring into the conversation via Skype Arash Izadkhasti, physician and infection specialist, who's been working for Doctors Without Borders since 2005, and just back from Kailahun, Sierra Leone, with working there with the Ebola outbreak. Welcome to Crosstalks.
Thank you.
Arash, how would you describe the situation in Sierra Leone? What have you seen?
Well, just to mention, it's been 6 weeks since I've been back, so, but I mean, I'm quite updated, and actually the situation has not changed much in Sierra Leone. I would say when I arrived, which was in the beginning of September, already end of June, the center had started accepting patients, and we had around 60, 70 patients, and then increased to 80. Confirmed cases probably around 60, 65, and then suspect and the rest. But the situation was severe. I mean, the situation was that we had to say no to 10 patients, approximately 10 to 20 patients per day. Sorry, I'm hearing an echo, but I guess I cannot do anything about it, right?
I'm afraid not, but we can hear you just fine.
Good. The situation that I arrived to was a bit difficult because of what you have to understand when you work with Ebola especially, which I didn't. I tried to understand it before I went there, but it's the fact that there's a fear factor that you don't really can imagine before arriving among people, among health workers, everyone you meet, which makes it quite difficult. And for that, I think and the desperation that was in the country, we had a lot of cross transmission among patients who were sent to us, who were not confirmed, who were not tested, because they put everyone, at that time, they put everyone who is, I think they still actually do that in Sierra Leone. I mean, the situation is a bit different in Monrovia, but in Sierra Leone it's still same. So they put 8 to 10 patients in one ambulance, and sometimes some of these patients are not confirmed cases. But in that ambulance, you have a lot of body fluids going on. People vomit, they bleed, or they have diarrheas. And I would say most of these patients, if there were cases who were not Ebola cases, would become Ebola cases. As it was mentioned before, I mean, it's a direct contact. So the fear basically is a bit strange. But the problem with Ebola, and what you realize, because I've been working in Sierra Leone before, is actually the fact that there is no proper medical healthcare system in the countries, and those who exist, there is not a good and solid rooted knowledge of infection control and basic, infection hygiene, which makes it quite difficult. As you said, as it was mentioned before, it's basically, I mean, it starts to spread via healthcare workers most of the times. And if you don't have a proper system for infection control, then it will be, a disaster like we have been facing now.
And that's why it's of such vital importance here to actually have foreign health workers there, because of the lack of the local system, and because of the lack of resources, so that you can put people in who won't spread the disease, because you will have the equipment you need and so on.
Exactly. I mean, it becomes a very expensive and very management-wise difficult infection control to handle. Even in our structures, this will be difficult, but then we are used and we know how to do, and we don't have that amount of patients coming to us. But in Sierra Leone, it becomes expensive to have the proper equipment. It becomes also a bit difficult because of the understanding of how it transmits. Actually, how you take on and take off your equipment and how you use it. You have to know. One of the things that strikes you when you arrived into, for example, when I arrived to Sierra Leone, is actually how people use gloves without understanding how it should protect them. Which makes you, if you use a protection that you don't know how to use or how you protect yourself, it actually becomes more dangerous, because then you're keeping that glove and then you use it to different things, and then you scratch your face, and then so it actually becomes more harmful to have protection that you don't use properly or take off properly than not having protection.
I find this very interesting, how much of this is actually a knowledge problem as much as an infection problem. Lars, you've also studied the Ebola outbreak with your mapping method. What have you been able to learn?
Well, if you look at the statistics from the WHO, I have visualized that in map sequences, and you can see that it started in 1976 in the present Democratic Republic of Congo near the Ebola River. And then there were also an outbreak in Sudan. And then it didn't happen much, or much was not registered anyway, or documented. But then in 1994 it started again, and then there were cases also in West Africa, in I think it was in Cote d'Ivoire, but none of the outbreaks before 2014 has been as dramatic as this one by far, and that's a question I also have to Arash here. Why, if you have any idea on why it is so dramatic right now?
Well, as everybody else who been involved in this would mention, if you look at where it spreads, it's actually a crossroad between, or kind of crossroad between. It's a meeting of the borders between Liberia, Sierra Leone, and Guinea. I mean, in the center where I worked in Kailahun, the distances between Kailahun and Gekedu are not far. Many of the people you meet in our centers had relatives on the other side in Guinea. So and before that, people would cross the border to go to each other's market. So I think it's actually because of the changes of and because the people who were this time affected are moving more and doing more, trading more with each other. And they are the same tribe. The Kisi tribe is both on the Guinea side and the Sierra Leone side. So I would say it's because of the movement.
Yeah.
Mm-hmm.
Sorry.
I have one
Absolutely.
more question here. You don't think it has anything to do with the travel patterns of the fruit bats? As one I've seen that they can travel very large distances, very far distances in a short time, like 300 miles in a few days or so. And it has nothing to do with the immunology of the bats, you think?
I'm not an expert on this area, so I wouldn't. But my guess is that it is not, because if that would be the case, then basically we would have, or maybe that is the case, because at the moment we don't actually know how many lines of spread we have, because you see the numbers decrease and then you see them go up. So actually the epidemiologists I know about are a bit confused, because now you see the numbers go down in Liberia. Before you saw the numbers go down in Guinea, but now it's increasing again and it's high again. And in Sierra Leone, actually, it's been high all the time. And in Liberia, now it's decreasing, but everybody's afraid that it might increase again. Anyway, I mean, if that would be the case, then why wouldn't it be? So why was it centrated in this area only, and then it spread? I mean, I'm sure that it actually it spread more by human beings, but when it comes to the bats, I can't really say, because I'm not really I haven't really studied that much.
We can step back a little bit from this specific case. Rob, I was wondering, I know the one thing that has changed through the years is that we can now actually identify the speed of which we're able to identify what causes infectious diseases. Could you elaborate a little bit on this?
Sure. I mean, with anything, when you talk historical facts or timeline, you have to factor in technology. And as far as infectious disease goes, it's actually quite a young field in terms of science. And one of the biggest breakthroughs was first being able to culture organisms or microorganisms such as bacteria. And then when we developed the capacity to culture these bacteria, we could then separate them and identify that the Black Plague was actually from a bacteria. When it came to viruses, there was a significant advance, which sounds quite simple these days, which was the development of the filter. So you're able to separate bacteria from other fluids, and then you could take the non-bacterial containing fluids and demonstrate that was actually an infectious agent. And at that stage, that's kind of what was used to identify influenza as a non-bacterial pathogen, which later became a virus. And then that's been followed up, I think, after about World War II with the development of the electron microscope. And the electron microscope lets you look at something on the size of nanometers to see the morphology. And so in terms of 1976, when Pieter received the blood samples from the Ebola River in his lab, they were able to directly take those samples, put them underneath the microscope, see what the virus looks like, and then just by similarity, they were able to say this looks like kind of similar to Marburg virus, but in fact it's different. And so then they were able to identify Ebola directly in the sample. And then more recently, we can directly sequence samples. And in terms of speed, we're talking decades to months to now weeks in terms of identifying what's causing
So
the
yes,
infectious
there
disease.
is of course a delay because somebody on the ground needs to realize that this isn't just an influenza, or this isn't just whatever the local.
It's not the normal.
It's not the normal, whatever it would be. Yes,
yeah.
And then you can actually get results relatively fast and put people and get a response team on the ground. But then already, of course, even very fast could be a month, 2 months after the outbreak begins. I'm wondering a little bit about these theoretical models that we've been talking about how much they can be used for predictions. So Peter, for instance, humans feel to me, just from engaging with them, as incredibly unpredictable. But can we use, statistically speaking, are we predictable? What can we predict that can help us mitigate diseases?
Yeah, this is the problem of all statistics, of course. The average person doesn't really exist. But so for the start of an epidemic, or also for the very end, individual traits of people are very important. However, when there are, say, more than 30 people infected, then the people infected start to become pretty average. Yeah. We have a good distribution or an overview of how people will behave, and then they are predictable. And sometimes we have to average in a clever way. Like a good example is sexually transmitted diseases, where you can ask how many partners does somebody on average has per year, or does the average suite has per year? Well, that's perhaps not a question you should ask. You should be more interested how many partners has the partner of an average suite, because if you have only one partner, or somebody has only one partner, there's only one way of getting infected. Yeah. Somebody has 10, then there are 10 ways of getting infected. So it's way more.
I'm realizing there's also a human factor, because sometimes you ask people about these things and they will absolutely lie.
Yeah, there are interesting data from sociology on that as well, like the number of heterosexual contacts on average per man is higher than per woman. There's something wrong there. But yeah, of course, we cannot get a very good idea of that. But still, our models are pretty robust against those fine details. So even if we don't get it exactly, or if we are 30% off, then what we observe and what we want to know is still pretty predictable.
Well, what about Lars? What can we learn about future pandemics from looking at the historical ones? Is there something? Are there rules that are emerging of what always happens, or what we should be doing?
Yes, looking at the influenza pandemics, you can see, at least on the cases that I've studied here in Sweden, that the epidemic period, that is when about 90% of all cases occur, is approximately 10 weeks. So you know that the influenza is seeded over many months, and then when the temperature starts falling, you can notice there is a strong correlation with the number of cases after you have had a steep fall in temperature. So that is something that you can learn. You can be ahead a little bit, and then also I've seen that, say, the geographic mean of the cases, they are moving along a line from southwest to northeast, and that's fairly typical. Knowing stuff like that about the typical behavior of pandemics can help authorities to be prepared in giving antivirals and vaccines.
There was actually on the news this morning, they reported that there had been some promising results with an Ebola vaccine. I don't know if any of you have heard about this or indeed read anything related so that you'd be able to speak about it authoritatively. But what would be the role strategically of a vaccine at this point? The pandemic is already happening. What would be, who would we start by vaccinating? I'm assuming health workers, but then what?
I mean, I think it's best for Arash to answer this, if that's possible.
Yeah. Do you have a
Arash,
feeling for that, Arash? What would, if we had a vaccine for Ebola now, what would we do with it first?
I think first of all, as you said, health workers who are in the first line to get affected should be vaccinated. I mean, it's already been kind of half experimentally used as a post-exposure prophylaxis. The problem, though, with the vaccine is that ethically you cannot really assure yourself how good is the. I mean, when you do the test on monkeys, then you can expose them to the virus. When it comes to humans, you cannot exactly do the same.
Oh, no. Yeah.
So still, we cannot really stop using the protection we are using, because also the whole equipment, the PPE that is used by the health workers is also not to spread the virus. I mean, not to transmit it to others. So even though if you have been an Ebola patient who has been cured and we know that they are immune at least for a certain amount of time, if they go back to work with other patients, they still have to wear the protection not to bring the virus with them out. And I think that will be the same even with the vaccinated health workers. The other thing that we could do is, of course, the neighboring countries and the regions who have not been affected, if there are any. I mean, in Liberia, definitely there are some regions that maybe haven't been really affected yet. The problem, though, is that at the moment in these three countries, Sierra Leone, Liberia, and Guinea, I mean, the normal healthcare and everything is a little bit. I mean, there's a fear of accidentally meeting Ebola patients. So to start a vaccination campaign will not be really, I think, that easy. I have to go back again to the as well. Sorry to interrupt, but when it comes to the fruit bat theory, I just have to say that I think. I mean, every night I was in Sierra Leone, I had at least 5 or 6 of these bats flying over my head, and I think if they would be the source of the transmission and increase, we would have a different way of being affected. And I don't really, because people stopped, I think, eating them as well. It's not that they eat it all the time or they get affected in that sense. So I just think this is actually something that should have been addressed a little bit earlier. You said, how can we predict something or learn something? And I'm going to be very politically correct here and say, well, what we could learn is that this affected the poorest of the poorest countries in the world. We realized that there is a problem, but we didn't actually pay attention until the problem became too close to ourselves. And I think that's one thing that we should learn, actually, to make sure that there is a healthcare system in even poorest countries and improve those to be able to defend this in the future.
Yes. Lars.
Yes, I was thinking along those lines too when I listened to an epidemiologist this morning, and he said that healthcare workers now fear Ebola so much that they don't dare be close to blood or body fluids, and that affects the normal healthcare. I mean, we're talking about countries where in the malaria belt, also here, and there are like more than 500,000 people dying a year in malaria, and 100 children a day. And these people, and I think also the pregnant women, so giving birth, they suffer a lot. So the effects of the Ebola is much worse than the virus itself can cause,
yes.
Or the bacterium, sorry.
Some people say that a big pandemic, something similar perhaps to the Spanish flu in impact, is about to come. And I don't know if this is an apocalyptic vision or a statistical probability. I think one of the reasons we responded so strongly to Ebola is that a lot of people, perhaps we've just been watching too many action movies, but we're very worried about pandemics, and we thought, well, maybe this is the one that's going to get us all. Now it looks like Ebola is not going to be the one that gets us all. But how likely is it to see, let's say in our lifetimes, the next 30, 40 years, a big pandemic that would have that big kind of impact like that, globally speaking?
I mean, I can start. I mean, I wouldn't venture to make any predictions as such, but I think as what we learned through history is the faster people travel, the more people interact, the more likely things are to spread and go fast. So things today can start in Australia and end up in Canada or the United States overnight. I think that's the real difference between today and the 1300s. So if Black Plague takes years to go by boats throughout the world, in 1918 we are still not traveling too quickly between continents. So it takes a few years to go or a year to go from one continent to another. But today it can happen really quick. But we also have more advanced technology in terms of diagnostics and more awareness that if somebody shows symptoms that I think are abnormal, people are going to actually look into those samples. And with these types of tools, maybe identify it faster. So hopefully those two kind of balance each other off, and that would prevent anything from happening. But to cancel the idea of surveillance, I mean, when Lars was talking about geographical positioning and knowing where, for instance, influenza goes from one place to another, if we continue to survey that where we think influenza kind of starts on a yearly basis, we can get insight into what strains are out there and then kind of analyze how deadly this current strain is. So I don't think we should abandon any surveillance techniques. So we should stay vigilant, I guess, is the right word.
But you're not very scared.
I don't know, life's unpredictable.
Very well, Lars, do you have a feeling about this?
I think that's something in some ways scares us all because these enemies are invisible and you don't know how to defend yourself. Say if it's something airborne, lethal, then you're in a bad position, and then we just hope that won't come.
Yeah, Pieter, what do you say?
Well, we haven't seen that many epidemics like this or pandemics like the Spanish flu in world history. So it can happen tomorrow. It can happen. That it lasts another 200 years. I think it will happen at some time. That at some moment, a disease will spill over from the animal reservoir to the humans, and then we are in trouble. And hopefully the medical science and the biology has some answers then.
And again, that we can have healthcare available also for the weakest in our society would be key as a preventive measure.
Yeah, that is a problem indeed, because we can make all our calculations, but if all of a sudden we need 1 million hospital beds, then we have still trouble.
Yes. I think it's time to let the audience in. I don't know, they're looking slightly terrified. In that case, maybe good, or maybe I apologize. Does anybody have a question? Okay, you think about that for a little while. Pieter, I have another question. A sort of follow-up. I know you've spoken before about chance or odds, how non-mathematicians don't grasp that very well. Could you explain what you mean and how we should think about the statistical chance to contract the disease?
Okay. Well, first of all, also many mathematicians have problem with grasping the concept of chance, especially in their daily life decisions. Like there are still mathematicians who buy lottery tickets. But the fear we have for Ebola, for example, might be one of those things. Like Ebola is something, once you have it, you're in big trouble. Still, influenza kills a lot more people on a yearly basis. And we are not that afraid of influenza. We probably, I'm coughing a bit, it's probably a little influenza. Well, probably you're wrong then, but still, we don't fear it. And also the way we treat, or some countries want to treat their people coming from Ebola-affected countries. Like Ebola is believed to only spread from people who are already sick. So isolating people or having travel bans wouldn't be too useful as long as we can trust that people who get sick after have been there report themselves and, well, perhaps not take the public transport going to the hospital while vomiting around. But there is not that much danger, as far as we can see right now.
Yeah.
And we only create fear if we take more control measures or more isolation measures.
And I realize, as you were saying, that of course this also goes for the sort of moral outrage that we feel that people aren't in the Ebola-stricken countries are not receiving sufficient care. Then of course we should feel that outrage for malaria as well, as you were saying, and that doesn't happen. That doesn't happen either to the degree that it should. Do we have a question from the audience? Yes. Please state your name and your question, please.
I am David. I'm studying in Stockholm University. And my question is, more than talking about what is being done now, what could have been done more in terms of prevention? Because we know, I mean, as any outbreak has this unpredictable quality to it, right? And so, because there are some guidelines, some strategies that WHO has outlined for for trying to prevent these situations, of course, before they happen, or I don't know, as other strategies as using like as I think Arash talked about post-exposure prophylaxis, like pre-exposure prophylaxis kind of treatment. I mean, what could have been done also in terms of surveillance, as you mentioned?
Well, that's very good. That's a very good question. From your different perspectives, who would like to start? Rob?
Arash, do you want to start?
No?
Or
Arash?
I could, actually. Thank you. Well, basically, when it comes to treatment or vaccination, as in post-exposure prophylaxis, let's face it, it couldn't be used. I mean, already we used what we had very sooner than we normally do. And the reason for it is simple. There is no money in this. And that's why we never developed any medicine against Ebola. When it comes to surveillance, definitely we could have done a lot more. But again, these are countries who are really, really poor. I mean, most of the health care system is based on help from outside, I would say, and in many of these parts or regions. And how it started and how it was, I mean, spread from the beginning, unfortunately, it did deceive us a little bit, since it was thought to be maybe cholera, because it was a lot more vomiting and diarrhea, and so we kind of discovered that it is Ebola a little bit too late, and then it took actually some time before we realized that it's getting out of hand. I think if we would have reacted already in, let's say, April, May, I would say, or even June, with more structured isolation of the patients and better contact tracing. Because what you have to realize is that even though MSF or Doctors Without Borders were there working, we didn't have enough or sufficient means to do a proper contact tracing and isolation of the suspect cases, unfortunately. So I think basically we could have done a lot more than what we did. I mean, and I'm not talking about only Doctors Without Borders, but WHO as well. So,
So basically,
I don't know.
What is existing methods, but more resources and acting faster?
Yes,
Would any of you like to add to that, Rob?
Sure, but I mean, it's really hard. If you think of surveillance, I think influenza surveillance is probably the one of the most up-to-date technologies in terms of looking at pathogen evolution. But we have to go survey in swine species or pigs. You want to check the bird population. So, I mean, basically, if you're talking Ebola surveillance, then you have to start looking at what type of virus is in the bats, how's it changing. It's an extremely massive undertaking in terms of financial resources. And
yes. We're running out of time. I should say, but that makes it an interesting frontier again for students looking for directions to go work. Unfortunately, that is all we have time for today. Crosstalks will be back on January 22nd with a discussion about one of humanity's most important challenges, the access to clean and sustainable energy. I would like to say thank you to Pieter Trapman, Lars Skog, Robert Daniels, and Arash Izadkhasti. See you then. Until then, be safe and be brave. Thank you.